Go ahead.
Okay, so welcome everyone, and I would like to thank everyone who is joining us from all over the world. Also, the AAPA for giving us the opportunity to gather and talk about this topic, and especially the speakers for sharing their time and knowledge with us today. And this is the first of a series of scientific webinars that the AAPA, the American Association of Physical Anthropologists, is bringing to us monthly. And I feel very honored to be the chair of the first webinar. And today's webinar we're going to have 3 speakers. Each one will speak for about 15 minutes, and then in the end, we're going to have about 15 minutes for questions. And the first speaker is Monica Green, who is an independent scholar and is going to talk about the new history of Black Death. Then we're going to have Dr. Luis Quintana-Musi from the Institut Pasteur and Collège de France. And he's going to talk about the pathogen-driven human evolution. Then we're going to have, in the end, Dr. Lucy Van Dorp, who is going to talk about patterns of genetic diversity in SARS-CoV-2, the agent of COVID, as you know. And I hope you enjoyed the webinar. And now let's hear Dr. Monica Green. Thank you very much.
I'm gonna jump in for just a second. Oh, no worries. To remind people how to ask the questions. So some of you have already figured it out because I'm seeing some questions. If you go to the GoToWebinar control panel and then click on the questions box, you'll be able to type in your questions and we'll be able to see them and we'll read them to the panel at the end. And if you want your name to be anonymous, because I usually announce the name of the person asking the question, just type anon at the beginning of your question and I won't read your name. Okay, with that, I'm going to, let's see, Monica, I'm going to prompt you to make you presenter.
Okay.
And you should be able to share your screen then.
Okay, can you see me?
Yep, it's perfect.
Okay, beautiful, thank you. Oops. Oh, and now it doesn't want to diminish the screen.
If it's not, is it not? Oh, you're trying to move the GoToWebinar box?
Yeah.
Try clicking on the little orange arrow.
Okay. Yeah, the orange isn't coming up. Now it's up. Okay, we're good to go. Good morning, everyone. Good evening, wherever you are. I am a historian, so I'm an interloper here, joining the physical anthropologists. And what I want to do is make a pitch for why history matters. And why particularly the history of this old disease matters, the history of plague. I am a medievalist. My specialty is as a historian of the Middle Ages. I'm also a medical historian. And as a medical historian, that means that I am also interested in the history of disease. And for the Middle Ages, the 2 most important and most socially significant diseases are plague and leprosy. And both of those diseases have been transformed in the understanding that we have of them because of genetics. And so that's why I am been an interloper into trying to figure out what is going on with the new genetics of these various infectious diseases, and how that new knowledge about the evolutionary history of these diseases changes what we need to do as historians, changes the questions we need to ask, changes the range of how we define our questions. And what I want to do today is tell you something very quickly about what has changed in the history of the Black Death. So the Black Death is a phenomenon I'm sure everybody has heard about because it has been a standard part of the narrative of European history, of Western civilization history, of world history for decades, actually for centuries. But a part of the problem with the history of the Black Death as it has usually been told, is summarized in this map. And this is a very common version of the map of the Black Death. And what it does is it starts in the region of the Caucasus, the area between the Caspian and the Black Sea. And then it shows this disease phenomenon that's coming in waves, moving across Europe, moving in the Black Sea, moving into the Mediterranean, and then moving all around North Africa and Europe. And as you can see also from this map, it has a very specific chronology. It starts exactly in 1346 and then persists up through 1353. And what I want to do is complicate both the geography of this narrative and the chronology of it. And I think both the chronology and the geography are much, much bigger. And also the biggest reason why we need to start revisiting histories like this in our current pandemic moment and thinking about what diseases are and how pandemics happen. So what we know about plague's history has, as I said, been transformed by genetics. This is the phylogenetic tree for Yersinia pestis, the bacterium that causes plague. This is a fairly recent version of the phylogenetic tree, and what it does is it shows major events. So there, all of those blue dots at the top of the phylogenetic tree represent genomes that have been sequenced from samples from the third plague pandemic. The third pandemic is the modern one. I live in Arizona, and we have plague here in Arizona because of the third plague pandemic. Essentially, plague got to Hong Kong, got on steamboats, and then moved out of Hong Kong around the world through these mechanisms of international trade that existed in the late 19th century. There in the middle, we have the Black Death, which is what I'll be talking about today. Below that is the Justinianic plague, which is usually called the first plague pandemic. So the Black Death begins the second plague pandemic, and then we have the Justinianic plague in the 6th to the 8th century. So at the very end of antiquity. And then this is just an example of one of the ways that genetics has transformed everything that we know about plague is it is put literally on the map the history of plague that we never even knew existed before. So there's a whole lineage of Yersinia pestis that is now part of what's called the late Neolithic Bronze Age lineage. So we never knew about that until the genetic work that's been done literally since 2015. Okay, so that's our big picture of Yersinia pestis. What I want to do now is focus in on just the middle of that story. And there's that phylogenetic tree again, and there's the center of it. That's the beginning of the Black Death phenomenon, the second plague pandemic. And what I want to talk about is actually just a part of the part, which is that right there. And what that is called the Big Bang. And I know it doesn't look very impressive the way that it's represented there, but the important point I want you to grasp immediately is it happens before the Black Death, before that narrative that we have so richly documented from Boccaccio and de Mussis and thousands of other sources that we have telling us about the experience of the Black Death, the massive mortality of that event. Something else happened before, and that's what we need to talk about. Okay, so the little bit of science I will indulge in here is just to invoke SNPs. A SNP is a single nucleotide polymorphism, and all it means is simply 1 point on the genome changing. And for the purposes of historical analysis, SNPs are important simply because they become permanent markers of genetic lineages. And that's the only point that I want to make about that here. Over on the right side of this diagram, we have a more schematic view of the phylogenetic tree that I was just talking about. And again, to stress, here's the Black Death. But the Black Death is not at the center of this larger phenomenon. The Black Death is just one part of this story. So the Black Death has been documented now in multiple samples from France, from Spain, from Sweden, from England, from Germany. The identical genome of Yersinia pestis has been found in various Black Death grave sites from throughout Europe. So we know exactly what the Black Death strain of Yersinia pestis was. Looking beyond that, we see Boulder City, and that is leading to branch 1B. So remember when I talked about the third plague pandemic, that will be the foundation for the third plague pandemic. And Boulder City is in Russia. Lyshevo is a genome that was just published last year, 2019, and that also is in southern Russia. But asking historically what's going on, the question then becomes, where was the polytomy? So the Black Death struck Europe, Lyshevo is in Russia, but where was the polytomy? The genetics actually doesn't tell us that yet because we have no AENA. But I, as a historian who is trying to put together some kind of meaningful narrative to explain this phenomenal disaster, has started asking questions. And so what I've been doing is taking evidence from the phylogenetics, taking evidence from the paleogenetics, and then combining that with both with field studies of the distribution of plague as can be documented from modern samples, and then also combining it with the historical documents that I normally work with as a historian. And this is what I found. So that question, where did the Big Bang happen? The indication of it, this is not proof, but the indication of it is that it is in a completely different part of Eurasia. Not Europe, not the Caucasus, but Central Asia. And so this is the area of the border between what is modern day China, Kazakhstan, Kyrgyzstan. And this is what we find, is that the distribution of the strain of plague that precede the first plague pandemic, the Justinianic plague, they're found in this region, the Zhongar Basin, in northwest China. After the Justinianic plague, we've now moved into an adjacent geographic area where we find 0-ant 2 and 0-ant 5. So those are created after the Justinianic plague. And then 0-ant 3, which is just before the Big Bang, has now moved into yet another adjacent geographical niche. So that's what the indications that the geography, the phylogeography gives us for where the Big Bang might have happened. So if the Big Bang happened in Central Asia, the Black Death happened in Europe, how did plague move around? Well, this is what I am arguing now, which is that it happened not in the 14th century, which is, as I said, the normal chronology we use to talk about the Black Death, but in the 13th century, a century and a half earlier. So what happens, and again, as a historian, I have a bias for looking for humans and what humans do. And what I see in the 13th century is the Mongols. The Mongols are the largest, they create, in a few decades, they create the largest land empire in human history. And what I am arguing now is that in the process of doing so, they are also distributing plague throughout Central Eurasia. So the Mongols emerge out of Mongolia in 1206. By 1209, they have allied with the Uyghurs of the Tarim Basin, which is right on the edge of the Tian Shan mountain range, which is where we just saw all those pre-polytomy strains of Yersinia pestis. By 1213, we get our first reports of Mongols bringing plague to China, of major Again, we cannot confirm from the descriptions that we have, but these are certainly plague-like events that are starting to happen in China. Plague-like events that start happening in the 1250s in Iran, in Iraq, in Syria. And so my suggestion now, and this is a study that will be coming out later this year, is that it's in the 13th century that all of this distribution happens. And much of it is under the radar, is that nobody is noticing or nobody's reporting, nobody's connecting these stories together. How did it happen? The Mongols are doing what the Mongols did, which is fierce and formidable military advances. What they're doing in the course of that is looking for food and looking for material resources. And what happens is a classic spillover event, is that the animals that now serve as hosts for those different strains of pre-polytomy plague are marmots, various marmots of the Tian Shan range and the region around there. The Mongols are using the marmots for food, they're using the marmots for fur, and they're using the marmots as a source of leather. So that's what gets them in contact with the marmots. Then what spreads, the marmots have a limited range, but what spreads the plague around is that the Mongols are also collecting grain, again, to feed their troops, one of which is a particular kind of millet that is grown right in the foothills of the Tian Shan range. So a spillover event from the marmots to other rodents is what I'm assuming. They pack up this grain in linen bags and are shipping it to all the sites of their sieges. And so that's why we get this phenomenon. In Chinese sources, in Persian sources, in Arabic sources, indicating that these outbreaks of these plague-like events occur at the end of sieges. Now we can look at the 14th century. Okay, thank you for the time. I'm just about done. Now we can look at the 14th century and see this explains the map that we saw earlier, is now we are looking at localized events. There have been climate shifts in the 14th century. This is the beginning of the Little Ice Age. There is a large-scale cooling that is going on in the Northern Hemisphere, and I am suspecting that there might be as many as 5 active theaters of plague that have now been seeded because of the Mongols' activity in the 13th century. Contaminated grain supply seems to be the consistent pattern of moving plague long distances. And then in the 15th century, we get new patterns of dissemination. The climate shift, at least in certain years, becomes even colder, and we get some new extreme long distance shifts of plague, moving the Russian strain of plague back eastward into China, and also, I also believe, into East Africa. So to conclude then, why is the Black Death a good example for what we need to think about now? We need to think interdisciplinarily. We need to think about all the different ways in which microorganisms, perhaps some kinds of animal intermediaries, and other factors pull things together and create the conditions of pandemic spread. And we need all of these different disciplines. We need historians, we need archaeologists, we need geneticists, we need modelers who can get us some information about the kinds of things we will never be able to reconstruct. In this case, I have no hope that we're ever going to piece together the history of the fleas that might have been involved in moving plague around. So I have many other things that I'd be happy to talk about in terms of the connections between the Black Death and other historical pandemics. I'll stop here. There's this brief bibliography. There's a very long bibliography available on Google Docs, and I'll post that link on my Twitter account. So thank you to Eduardo, to the AAPA, for the invitation, and I'll now turn things over to my other colleagues. Thank you.
Great, thank you. Luis, I'm going to prompt you to share your screen. Oops. Okay, it should be prompting you now.
Okay. Can you see that?
Yes.
Okay.
Perfect.
So thanks to Eduardo and to the AAPA for inviting me to tell you a bit about how we can use population genetics to better understand not only human evolution, but in the context of these seminars, to understand the history of human exposure to some infectious diseases, and also how other human forms, such as Neanderthals or Denisovans, may have helped early modern humans to adapt also to pathogens and to infectious disease risk. So, oh, something is going on.
If you click on your presentation, just click in the middle of your presentation, then it should allow you to advance your slides.
Great, thank you very much. So as you all know, humans, Homo sapiens, left Africa around 60 to 80,000 years ago to colonize the rest of the world. But in the meantime, early Homo sapiens met other species or other forms of humans, such as Neanderthals in West Eurasia and Denisovans in East Asia and the Pacific. And during these dispersals, humans have to adapt to a large variety of environments, including different climates, different nutritional resources, but also different pathogens to which they had to resist. So today, in the next 15 minutes, I'm going to tell you about the relation between archaic introgression, meaning admixture with ancient humans, in the context of immune functions. And we're also going to review two recent examples of my lab, in which we have a study using population genetic approaches, the history of human exposure to two major infectious diseases, namely malaria and tuberculosis. So thanks to ancient DNA studies, we know today that during the dispersals of modern humans in Asia and Europe, they get mixed with other human forms. And today in the genomes of non-Africans, we have around 2% of Neanderthal ancestry. And in the genomes of East Asians, and particularly in the genomes of Pacific Islanders, we can find ancestry that comes from Denisova to a range that can go up to 5%. Of course, these are averages across the genome, because if you walk along the genome, you can find regions, specific genes or genomic regions that show up to 65% of archaic ancestry. We know also that in general, this introgression of archaic material into modern humans was selected against, suggesting that the first hybrids between Denisovans and modern humans, or Neanderthals and modern humans, were not super successful. But having said that, there's regions of the genome that show high scores of archaic ancestry, and in particular, evidence of adaptive introgression, meaning how this introgression of archaic material into modern human genomes provided some kind of selective advantage. So this is a question that we started to address a few years ago in the context of innate immunity. Why innate immunity? Because, as you know, innate immunity is the first line of host defense against pathogens. When you are infected, there are cells that comes with receptors, and these receptors, for example, are going to recognize that there is a pathogen that has got into your body. So the question we wanted to address here was how and if innate immunity genes are enriched in Neanderthal ancestry with respect to the remainder of the genome. Because if innate immunity genes are enriched in Neanderthal ancestry, this means that early Europeans, for example, through admixture with Neanderthals, they acquire adaptive alleles of innate immunity genes that allowed them to better survive infection. So for that, we interrogated the amount of Neanderthal ancestry of around 1,500 genes involved in innate immune functions, receptors, adapters, effectors, etc. And we compared the scores of Neanderthal ancestry with the remainder of the genome. And the answer was yes. Genes involved in Neanderthal ancestry are enriched, genes involved in innate immunity are enriched in Neanderthal ancestry with respect to the remainder of the genome, and this for both Europeans and Asians. Here you have an example of a genomic region that is strongly enriched in Neanderthal ancestry. Is the TLR1-610 region, encodes for receptors that sense pathogens on the cell surface. And here there is around 80 Europeans, and each line is an individual. And when you see a blue line, means that the 3 genes are completely encompassed by a Neanderthal haplotype. And you can see how there is a strong enrichment in Neanderthal haplotypes around this region. More generally, we identify around 80 genes in Europe and a non-overlapping around 18 genes in Asia of innate immunity genes that presents among the highest scores of Neanderthal ancestry in the human genome. So the next question we wanted to address here was, well, let's forget about genes, but let's focus on mutations that are involved in gene regulation. They are known sometimes like EQTLs, for expression quantitative trait loci, are mutations whose variation is associated with changes in gene expression. And basically what we show is that regulatory variants are enriched in Neanderthal ancestry, as you can see here, but specifically regulatory variants that are involved in responses to viral challenges, suggesting that there has been archaic introgression that has been beneficial for early Europeans to fight viral challenges. Here you have an example of a gene that is called PNMA1 that is strongly enriched in Neanderthal ancestry, and the more this gene is covered by Neanderthal haplotypes here in red, the more this gene is going to be expressed. Moreover, this gene is known to be involved in the recognition of influenza virus, suggesting overall that Neanderthal introgression has affected primarily responses to viral challenges. And this has been confirmed also by the group of Dimitri Petrov and David Enard at Stanford, that they showed that VIPs, meaning in this case viral interacting proteins, are enriched in Neanderthal ancestry. So talking about archaic ancestry, we have more recently focused on South Pacific populations. And why South Pacific? Because these populations present the highest combined levels of archaic ancestry, if we combine together Neanderthal and Denisovans. And for that, what we did, we sequenced the whole genomes of around 300 individuals from this transect that is thought to be at the baseline of the peopling of near and remote Oceania, starting from Taiwan, the Philippines, the Solomon Islands, Vanuatu, and also we include data that was from Papua New Guinea and the Bismarck Archipelago. So the first thing we observe in this basic principal component analysis is that near and remote Oceanians lie in between Papua New Guineans here in purple and East Asian populations. And all this line basically is just showing the different levels that these populations have of a mixture with Austronesian, East Asian related populations. But to come back to what interests us today is the level of archaic ancestry in the Pacific. And what we found is that Neanderthal ancestry is very homogeneously distributed across the Pacific populations, ranging from 2.2 to 2.9%. Moreover, when we focus on what are these genes enriched in Neanderthal ancestry involved in, we show that these genes are enriched in multiple phenotypes, ranging from immunity, neuronal development, metabolism, and pigmentation phenotypes, suggesting that Neanderthal introgression was beneficial for this kind of phenotypes. But in strong contrast, what we find is that Denisovan ancestry is very heterogeneously distributed among Pacific islanders, ranging from almost zero in Taiwanese Aborigines or some Filipino populations to 3.2% in Papua New Guinea, and in particular in populations from the Vanuatu. And more surprisingly, what we show is that most genes that are enriched in Denisovan ancestry are almost uniquely enriched in immune phenotypes, suggesting that early Southeast Asian populations, by admixing with Denisovans, they acquire advantageous alleles that they allow them to better resist infection and more generally infectious diseases and local pathogens. So let's turn now to more specific examples of how population genetics can help us to better understand the history of human exposure to infectious diseases. And let's start with malaria. Malaria, and more specifically, the sickle cell hemoglobin S mutation, is an iconic case of balancing selection. And why that? Because the sickle cell mutation at the heterozygote state protects against falciparum malaria, despite the price to pay for that is that at the homozygote state is associated with life-threatening sickle cell. So the rationale we applied here is that in view of the deleterious nature of this mutation, if malaria was not there, the mutation would be purged out of the population almost immediately in 12th generation. So dating the time at which the mutation, dating the time of the mutation actually is almost dating the time at which malaria was started to be a burden in the population, because if not, the mutation would have been quickly removed out. So this is what we did, and for that we sequenced the hemoglobin gene in around 500 individuals of different African populations from both West Central Africa and East Central Africa, and in particular different groups of Bantu-speaking farmers and rainforest hunter-gatherers, also known sometimes by the derogatory term pygmies. So we apply an approximate Bayesian computation approach. And basically what we found is that the sickle cell mutation appeared in the ancestors of what's today Bantu-speaking agricultural populations around 25,000 years ago, pushing back to a few thousand years the presumed origins of malaria in Africa. But more interestingly, what we found is that the mutation that appeared in the ancestors of agriculturalists was transferred to rainforest hunter-gatherers around 6,000 years ago. So following the so-called poison antidote hypothesis, farmers started to do deforestation, started to meet pygmies that they were living in the rainforest of Africa, and they brought malaria to the rainforest, but also they brought the mutation that they allow rainforest hunter-gatherers to survive malaria, that is the sickle cell mutation, providing a very good example of something that is increasingly known now in humans, that is adaptive admixture. And to finish, let me share with you our most recent results about tuberculosis. Tuberculosis, you know, that is is one of the three big killers in the world today: malaria, tuberculosis, and HIV. And the genetic basis of tuberculosis, at least the genetic basis of common tuberculosis, were until 2 years ago practically unknown. And the group of Jean-Laurent Casanova at the Rockefeller Institute, they identify a mutation, a common mutation in the population that is associated to increased risk to develop clinical forms of TB. And it ranges from 2 to 4% depending on the population. And as you can see here, is mainly restricted to population of European ancestry or populations that have also, due to admixture, European ancestry. Watch out here, the pie chart, sorry, the pie chart does not represent 25%, but represents 2.5% in the population. Sorry again. So this mutation is in a gene that is called TYK2, and the mutation is called P1104A. So we wanted to learn more about the evolutionary history of this mutation. And to do so, what we did was to study a collection from David Wright's lab of 1,000 ancient genomes that cover the last 10,000 years of European history, going from the Mesolithic to the Middle Ages and the present times. And what we saw is that the mutation appears from the first time in Anatolia around 8,000 years ago, to then decrease in frequency again, and increase in frequency until almost 10% during the Bronze Age, following the arrival of steppe-related populations associated with the Yamnaya culture, to then drop back in frequency again until its present-day frequencies of around 2 to 4%. So to understand if these marked fluctuations in frequency are compatible with simple genetic drift or negative selection, in particular this drop since the Bronze Age until today, we apply again an approximate Bayesian computation approach. The first thing we saw was that the mutation appeared in the common ancestor of West Eurasians 30,000 years ago. But this mutation appeared therefore 30,000 years ago after the split of West Eurasians with East Asians, but before the split of West Eurasians into European, Middle Easterners, and Central Asians populations. But most importantly, what we saw is that this decline after the Bronze Age is compatible with strong negative selection, starting 2,000 years ago, with a selection coefficient of 0.2 that is among the 3% most strong of the human genome, suggesting that tuberculosis have imposed a very heavy burden on European health over the last 2,000 years. Now we're applying this approach to the whole genome and to other genes involved in innate immunity to better understand which have been the main actors, either because they have been positively selected or because they have been negative selected, meaning advantageous mutations or risk-associated mutations, using a population genetic approach and using also ancient DNA over the European history and over the last 10,000 years. So with that, I would like to thank the main actors of all the things that I've been telling today, in particular Maxime Rotival for the first part of Neanderthal ancestry, and Mathieu Deschamps, Javier, Jeremy, and Sebastian and Jeremy, and Lara for the Pacific team, Guillaume for the tuberculosis, and Gaspar for malaria, sorry, the other way around. And thanks you to all for your attention. Thank you.
Thank you very much. Lucy, I'm going to prompt you to be presenter now. Okay, it should prompt you.
Does that look okay?
Yes, perfect.
Yes, perfect, great. So thanks everyone. It's a great honor to be able to present over this virtual format today, and I think it's nice to have a follow-up talk to the kind of historical studies we've just been hearing on malaria, plague, tuberculosis, and to bring us right back up to date to another pandemic that humanity is facing, which is of course COVID-19. So I'm going to talk about some of our work exploring or how we can use genomic patterns and genomic approaches to understand more about the evolution of the virus responsible for COVID-19, but also to understand more about how this pandemic is spreading and interacting. And I'll start with a timeline, and it's one that we're all now very familiar with. So at the end of last year, a cluster of viral pneumonia cases of unknown origin were identified in China. And these cases rapidly spread, resulting in an epidemic throughout China, followed by increasing cases around the rest of the world. By February of this year, the World Health Organization designated this as a brand new disease, COVID-19, which stands for Coronavirus Disease 2019, and a global pandemic, meaning we had sustained human-to-human transmission in many countries around the world, was declared on the 11th of March. And I would argue that a particularly important point in this timeline was the generation of the genome of the virus responsible for COVID-19. And this virus was sampled on the 26th of December of last year from a patient in Wuhan in China. The genome was sequenced, assembled, annotated, and made publicly available by the 5th of January of this year, which I think is really remarkably quick and has offered us the opportunities to really use genomics in the kind of pandemic response. And having this genome already allowed us to confirm that this was a viral pathogen. It wasn't bacterial, fungal, and in particular a coronavirus. Here's a big family tree of coronaviruses, and you can see that we can place the genome responsible for the virus responsible for COVID-19 into this tree, finding that it's a beta coronavirus closely related, or about 80% identical to that causing the 2002-2003 SARS epidemics. And so this resulted in this new virus being designated severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. And you can see from this tree that some of the closest relatives are those that are found within Rhinolopus, which is a genus of bats, of horseshoe bats in particular. And this is our closest sequenced relative. And looking across this family tree, we can see that coronaviruses are really found in a very wide range of mammals, and in all likelihood, SARS-CoV-2 jumped from an animal reservoir, which we're currently working to identify, into the human population, giving rise to our ongoing pandemic. So what does the genome of SARS-CoV-2 look like? Well, it's about 29 or nearly 30 kb or so long, which is quite large for an RNA virus. But the genome structure is broadly very similar to these other coronaviruses I showed previously. So a really large fraction of the genome is taken up by this first open reading frame in yellow. I don't know if you can see my pointer, but ORF1AB. And then about a third or so of the rest of the genome is taken up by structural proteins. The spike protein in particular, which you'll see from this little diagram, gives coronaviruses their characteristic crown-like or spiky appearance, which is where they get their name. And this is an important protein for binding and invading human cells, as well as other structural proteins, including the envelope membrane and nuclear capsid protein. And then towards the end of the genome, we have a real suite of accessory proteins, which is also what we observe in other coronaviruses, though some of these are unique to SARS-CoV-2. And so having this genome and instantly identifying that we have a brand new coronavirus in circulation has really allowed us to make large-scale genomic comparisons during the ongoing pandemic. And really the speed of generation and sharing of SARS-CoV-2 genomes has been completely unprecedented. The largest repository by far is GISAID. I've put their logo up here, where really labs all around the world are generating and uploading sequence data in what's pretty close to real time. So this plot gives you an indication, I guess, of the number of genomes being uploaded to this server since that first genome was made available at the beginning of January. And you can see really there's been a very steady and well, really quite fast increase in the number of genomes that have been available. So I made this plot this morning. I'm in London, so it's evening for me, and this corresponds to over 83,000 of these viral genomes sampled from countries, or over 100 countries all around the world. And you'll see that there's been a really large sequencing effort in certain regions, in particular Australia, the US and the UK, and that's largely thanks to the efforts of large sequencing consortiums here in the UK, particularly COG UK, who've generated or uploaded data for about 35,000 or so viruses. And we can use this really large data set, and in terms of data, this is quite an amazing resource. We have data points almost at every stage of this ongoing pandemic, and we can ask questions about how genomic diversity has emerged in this virus over the course of the pandemic. And one way of representing this diversity is as a phylogenetic tree or a family tree of the virus, and I've put up one of our most latest trees here, which corresponds to 43,000 or so genomes. And one point to note straight away is that actually not much diversity has accumulated. So Monica gave you an introduction to SNPs, and if we look at the x-axis here, we can see the number of SNPs that have accumulated from that first genome that was sequenced within China. And you can see that there's no virus strain which has more than, say, 35 mutations or SNPs from this initial or first genome. And this highlights to us that really this is quite a young virus, and also the fact that it's fairly homogeneous. So Given this is a 30,000 or so base pair genome, 30 mutations is not many. And in fact, if I take any two viruses from this phylogenetic tree and compare them, on average, there may be 8 to 10 mutations apart. So for all intensive purposes, we're dealing with one type of virus that's in circulation around the world. But nonetheless, we can use these small changes to infer some properties of the pandemic. And in particular, we can annotate this phylogenetic tree by the place in the world these viruses were sequenced. And the first thing to note is, of course, there's many colors where we're dealing with a global pandemic, but also that these colors are not necessarily structured. So we don't see what you might call strong population or geographic structure in SARS-CoV-2. And by that, I mean, we don't find that a virus that's sequenced in any one place looks exclusively like other viruses sampled within that place. And instead, it's really quite diverse. So to make that point a little bit more clearly, we can zoom in on, for example, this large database that we have of genomes within the UK and just annotate this family phylogenetic tree using those genomes. And you can see that essentially, if I take a virus genome within the UK, it has the potential to fall anywhere across this phylogenetic tree. So any one genome can recapitulate some of the global diversity. And this is really consistent with there being very many introductions into the UK. And in fact, others have more formally estimated this, detecting at least 1,300 distinct transmission events into the UK, which have then seeded local transmission chains. But this also highlights that kind of narratives around, for example, a patient zero or single index cases don't really hold true. And at least in the UK, we can see that there's been many distinct introductions occurring at many distinct times. And I've shown you the scenario in the UK, but this is not unusual, at least for the countries or regions where we have intense sequencing. So here's the same situation within the USA, and you can see that, again, any viral genome really has the potential to fall anywhere on this phylogenetic tree. Similarly, if we look at Australia. And so the overall picture is that although we have a fairly homogeneous or similar virus in circulation, we also have a landscape of diversity whereby everything is almost everywhere. There's kind of perhaps exception to this is if we look at China, and here I've just highlighted some of the genomes available from China, and you can see they largely fall into these two basal clades of the phylogenetic tree. And whilst this is fairly consistent with an age and origin, it's also possible that this reflects something of a temporal rather than geographic signal, with virus genomes sequenced in China tending to be from earlier on within the pandemic. So we can really see using the genomic data that we have extensive transmission, we have extensive introductions to many different places around the world. But we can also track the accumulations of mutations as a function of time to make predictions on when we think SARS-CoV-2, this new coronavirus, jumped into transmission in the human population. And to do so, we use a kind of simple premise, which is that evolution or mutations acquire as time goes on. And in this case, we have a data set of 43,000 or so viral genomes sampled over, say, 6 months. And this offers us opportunities to calibrate a phylogenetic tree just like this one by time. And in particular, we can ask the question, well, when do all of these 43,000 or so viral sequences, when do we predict that they last shared a common ancestor? And this common ancestor would correspond exactly to when we would predict the host jump occurred from an animal reservoir into human circulation. And so if we do that across this 43,000 or so genome database, we find that we land on an estimate of around 2019.86, which corresponds to around about the 10th or 11th of November of last year. And this again highlights that we're dealing with a virus which has been in circulation really quite recently. And in particular, we did a kind of smaller scale analysis. These analyses are fairly computationally intensive on a smaller subset of genomes, which allowed us to estimate a window of opportunity when we think SARS-CoV-2 jumped into the human population. And we currently estimate that to the early part of October of last year through to maybe early December. So certainly towards the end of 2019. And this is, of course, an important parameter to know. We need to know how long this virus has been in circulation, but it also tells us about how much immunity or baseline immunity we might expect within the human population. Of course, this also highlights that as a new disease and a new virus, SARS-CoV-2 was picked up relatively recently. And so we've seen how we can use mutations or SNPs to track back the origins of pandemics, in this case, SARS-CoV-2. But we can also use mutations or genomic diversity to learn more about some of the features of SARS-CoV-2, and in particular to identify potential regions under selection or adaptation within the virus genome, or those that we might want to target using, for example, drug and vaccine initiatives. And there's really been incredibly intense debate and interest in mutations as they've emerged in SARS-CoV-2, despite the fact that mutations are really a natural byproduct of evolution. However, we can use mutations to identify regions that are putatively under selection, those that may be invariant or offer the most hopeful targets, for example, for vaccines, but also those mutations which might be having an actual functional impact on the virus we're currently dealing with in circulation, perhaps changing its virulence or changing its transmissibility. And this has attracted a lot of media attention, with lots of kind of scary headlines about mutant viruses taking over the world. And one question we wanted to ask is, well, can we look at the mutations that we see being acquired within SARS-CoV-2, and can we formally test whether there's any impact or difference that they have on the virus transmissibility? And so to do so, we look specifically at mutations and their location along the SARS-CoV-2 genome. So this is now based on an alignment of nearly 47,000 genomes. And as I'm sure you're aware, this is a rapidly changing situation. And as such, we have an online app which allows you to kind of screen the alignment. But you can see that really there's the potential for SNPs or mutations to occur anywhere along this alignment. But there are some regions where we see very high frequency mutations. And you can see the 4 peaks, for example, within this alignment. But trying to disentangle whether a mutation such as those 4 or any others reach a high frequency because of demographic processes or because they're actually advantageous to the viruses is non-trivial. And so we could have a scenario where a virus acquires a mutation randomly during replication. It's introduced into a region where there's perhaps not much public health interventions. It spreads quite freely, and we see that mutation go to high frequency without it modulating the functional biology of the virus. Or we could have a mutation that actually makes a change that offers some advantage to the virus, and that might go up in frequency due to positive selection. And so we want to disentangle these 2 scenarios. And the way that we've currently gone about doing this is to focus on those mutations that have arisen repeatedly and independently given the phylogeny. And we refer to these as homoplasies. And currently, we think these are the best candidates for ongoing selection or adaptation within the SARS-CoV-2 genome as it's adapting to the new human host. And our analysis to date identifies 9 or 5,000 or so of these homoplasic mutations that are putatively adaptive, of which we can get down to a data set of 400, which we think are strong candidates. And the next question is, do any of these 400 or so mutations have any impact on virus transmission? Do they increase the transmissibility of the virus we have in circulation when it carries this mutation? And a convenient really property of working in phylogenetics is that really for a virus, the transmission fitness, or how well it spreads, can be considered as a proxy for the overall fitness. And this allows us to estimate, using a phylogeny, the impact of a mutation from an ancestral node to its descendants by looking at the number of progeny from that ancestral node. And so if I take this tree as an example, here we have a parent node, we have a red mutation that's accumulated. Again, I don't know if you can see my cursor, but this is a homoplasic position, so we observe it in 2 places within the phylogenetic tree, and we can ask the question, well, do we see more descendants carrying this mutation relative to the sister node that doesn't carry that mutation? So in this case, we'd say actually this red mutation appears to be beneficial. We see more descendants carrying red than we see white. And so we devised a statistic, which we call the ratio of homoplasic offspring, or ROHO statistic or index, which we could apply across this 46,000 or so SARS-CoV-2 genomes to ask the question, well, do any of these 400 mutations have any impact on virus transmissibility? And in the interest of time, I won't go into the full details, except to say if we zoom in on 2 in this scenario, again, you have a picture where we see more descendants carrying red compared to those carrying white. So in this case, we devise our ROHO score would be 4 carrying red divided by 2. And so it follows if we have a mutation which has no impact on viral fitness or transmission, we'd expect on average to find that this ROHO statistic is equal. And so what do we actually observe in SARS-CoV-2? And here's a plot along the genome with the line of 0 marking out that where we would expect to see no effect on neutral evolution. And here's our strongest candidates or mutations that we observe in SARS-CoV-2. And so the immediate thing to note is they all fall around this 0 line. And in fact, none of these mutations here or these scores that we've calculated show any significant change from what we'd expect under a completely neutral model. And so this really highlights that to date, none of the mutations that have arisen within SARS-CoV-2 have a very significant impact on transmissibility, which I think is so far encouraging. But of course, this is a situation that we're going to continue to monitor. But the other question is, well, do we see any other properties of these mutations in SARS-CoV-2? We have a lot of these recurrent mutations. Where do they arrive from? And to test this, we observe the changes that we observe in mutations. So what nucleotide changes do we see from A, T, C, or G? And quite strikingly, if we look at mutations in the SARS-CoV-2 genome, we see quite strong biases. And in particular, about 57% or so of mutations derive from C to U changes. And this is really quite asymmetric, and it implies that it's not just the action of the viral RNA polymerase during viral replication that's inducing mutations, but something else is going on. And to date, and it's kind of one of our hypotheses, and certainly one that's been put forward by others, is that human RNA editing systems have actually been responsible for many of the mutations we observe in SARS-CoV-2. And strong candidates here are the AID and APAPEC family of cytidine deaminases, which catalyze the deamination of cytidines to uridines. And really adding, I guess, some evidence to this point is that if APAPEC deaminases tend to preferentially target certain sequence contexts, certain motifs within the alignment. And certainly if we look for the mutations that we observe in SARS-CoV-2, and this is every possible of combination of 3 letters, we see an enrichment in those which have a C in the middle position. And this is a known target of APAPEC3A-like family of proteins, and suggests that we have kind of innate immunity interacting with the SARS-CoV-2 genome. And of course, the functional meaning of this interaction is really an ongoing question. It might be that our immune system is inducing mutations designed to render the virus ineffective, perhaps inducing a premature stop codon or otherwise deleterious mutations. But it also could be that these mutations may offer some fuel for evolution to act upon. But nonetheless, it's very clear that the mutations that we observe in SARS-CoV-2 are not just those induced by the virus or occurring from the virus itself, but also those that have been induced by our own interactions with us, its human host. So I don't know where I am for time, but in summary, large-scale genomic data, and it really is large-scale from SARS-CoV-2, is offering huge opportunities to infer or understand pandemics in a way we just haven't been able to before, in particular questions of origin, transmission, and spread. Our analyses really point to the fact that SARS-CoV-2 jumped into the human population towards the end of last year before it began to circulate really quite rapidly and quickly in human populations worldwide. Most countries, and I highlighted the US, the UK and Australia, but it really seems to be the same for many regions where we have a lot of viral sequences, have experienced many thousands of introductions. There's no patient zero. But at the same time, the virus that we're facing worldwide is really very, very similar. There's essentially one strain in circulation. Mutations are a natural outcome of viral replication, but of course they should be closely monitored. And in particular, we're focusing on mutations that have appeared repeatedly and independently. But to date, none of these recurrent mutations show a significant change in the transmissibility of SARS-CoV-2, though we have observed these marked base asymmetries, which are pointing to the role of our own human RNA editing mechanisms in shaping the evolution of this virus. And so I should point out this work is really the product of a huge team effort, and we've all been chatting over Zoom like nobody's business to kind of work and try and keep up with the rapid scale of sequencing across the course of this pandemic. So you can find a few more details here, and this is work that I've co-led with Francois Ballou, based at UCL Genetics Institute. Thank you.
Great, thank you, Lucy. I'm going to take back control of the screen. So we're right at the top of the hour. I think what we're going to do is we will invite the panelists to come back, and hopefully you guys can stay for a couple of minutes. I'll try to get a question for each of you from our attendees. There was a lot of chatter in the questions box, and people really enjoyed the presentations. Let me mention really quickly, our next webinar is going to be on the 15th of September at noon Eastern, and it's a community discussion about AAPA's 2021 annual meeting. So we'll be sending out information on that. You can also see it at our webpage, physant.org, and check out that. And the recording of this meeting, of this webinar, will also be available on the webpage, physant.org, in the next couple of days. It usually takes about 2 days to get everything up. Okay, let me ask a couple of questions. So this first one is from Maria Nieves Colon. It says, question for Monica. Great talk. have there been any DNA studies from the Middle East that have found Yersinia pestis DNA from the time period predating the Big Bang?
Can you hear me? Yes.
Yes.
The answer is no, and that is, oh my gosh, that is the huge terra incognita for what is going on in plague studies right now. ADNA always relies on archaeological work to find burial sites, to retrieve burial sites. All the ADNA that has been sequenced thus far has come from humans. So we have no animals, no rats, no, certainly no fleas. I doubt we will ever have fleas. So there's a very heavy Eurocentric bias of the data that is available now. And this is why, for me, adding in the phylogenetic work, the work that's been done from modern samples, from modern isolates of Yersinia pestis, has been transformative. Everything we know about plague will be transformed once we have samples from the Middle East, once we have samples from Asia, once we have samples from Africa, as I think we will eventually. So the immediate answer is no, and the hopeful answer is hopefully soon we will have some. Thank you.
Great, thank you. The next question is for Dr. Quintana Mercy. It's from Wynn Meyer, and it says, is there evidence that the tuberculosis risk allele may have been selected for in the population in which it arose, or was its increase to somewhat moderate frequency most likely due to drift?
Oh, that's a not easy one, because there's a lot of, we know very little. We know that there might be also, okay, this mutation might also, in any case, mutations in the gene, in the same gene, TIC2, have been to some extent associated also with protection against some autoimmune diseases, what may suggest that to some extent there could have been some increase in frequency due to positive selection. From a population genetics perspective, we don't found any evidence, first. And second, in general, autoimmune diseases in general are too late onset to be selected for, right? So the short answer is no. Yeah.
Thank you. Okay, so the last question, or this question anyway, is for Dr. Van Dorp, and it says, it's from Joseph Parrish. It says, if I'm interpreting Dr. Van Dorp's presentation correctly, the genetic diversity of COVID-2 is higher outside of China than inside of China. When we see species originating in certain regions and spreading out through the world, the greatest diversity is usually found in the place of origin. Does this suggest that China is not the likely site of origin for this virus, or is there something else at play that is operating at an informational level?
Cool. Thanks for the question. So one key observation we should make dealing with actually any genomic data sets, and SARS-CoV-2 is perhaps a good example of this, is that there's potential biases in what we're sequencing and observing. And in the case of the observation that we have the two basal clades in China and then perhaps less diversity is perhaps based on the fact that we've done there's been genome sampled in China early on in the pandemic and then made available and then less so further on. So we are observing now there's been repeat outbreaks, for example, in Beijing, and those viral genomes are falling into the rest of the diversity on the tree. But we just don't have a very good representation of the data that's there yet. So this is also a very plausible scenario. And I think all kind of evidence does support at least an Asian origin of SARS-CoV-2.
Great, thank you very much. I think we're going to end it there. We're having people drop because people plan around the hour. So thank you guys very much. This was a really great presentation. Eduardo, I don't know if you wanted to say anything at the end, but if you do, jump on. If you don't, I'll just thank you guys very much for a really great presentation. Like I said, we had over 400 people attending at one point, so it's great.
Yeah, I just wanted to say again, thank you very much for the speakers and for all the audience as well. Thank you very much.
Great.
Okay. Thank you
Take
all.
care, everybody. Thank you.
Thank you.